We have seen that Hox genes are part of the genetic toolkit for development in Drosophila. And what Hox genes do is they determine the identity of segments. And when you mutate the Hox genes, that leads to homeotic transformations. That is, one segment gets transformed to the identity of a different segment. Furthermore, we know that the way Hox genes determine the identity of segments is that, first of all, they're expressed in the segments whose identity they control. So the genes are being transcribed, and there's mRNA, and that mRNA is translated into protein. Second, these proteins encoded by the Hox genes are in fact transcription factors and contain this domain, this part of the polypeptide sequence called the homeodomain that binds DNA. And so the Hox genes act by being expressed in the segment whose identity they control, and then binding to DNA and regulating all the genes that give a particular segment its identity. For example, all the genes that make halteres, or all the genes that make wings, and so on and so forth. One question that remains unanswered is, how do Hox genes know what segment they are in which segments to be expressed? In other words, what controls the expression of Hox genes so that they are expressed in the right segments and not expressed in the wrong segments? It turns out that there is another set of toolkit genes called the anteroposterior, or anterior to posterior segmentation genes, AP segmentation genes. And it is the AP segmentation genes that in fact instruct the Hox genes to be expressed in the correct segments. The AP segmentation genes were discovered through a mutant screen. And what a mutant screen is a way of generating lots of mutant lines of whatever organism you happen to be studying, in this case fruit flies. Since it could, if you wait for mutants to arise naturally, it may take a very long time. Furthermore, as we discussed before, developmental gene, most mutants are going to cause lethality, and those embryos will die during embryogenesis and will never reach adulthood. So you may never even observe many mutants because of the embryonic lethality they cause. So an EMS mutant screen allows you to generate mutants, and the way you do it is that you feed fruit flies this chemical called ethyl methanosulfate, and EMS is a mutagen and causes mutations, and then you mate those flies and their progeny have mutated genes, and then you can go and screen, which means you can basically go and look through all the different mutant progeny that have been generated to identify ones that are mutant in a particular phenotype that you're interested in. In this case, we are interested in the body plan of the Drosophila larvae. And so you would go and look through the mutant individuals one by one in order to identify one that has something different, a different phenotype from wild type. And once you have found such a mutant, you will establish a line of flies that have this mutation. And since these mutations are usually lethal, you maintain these. They're not pure breeding lines, but instead you keep these lines in a heterozygous state. And just to kind of review the morphology of these larvae, because we will need to understand the morphology in order to understand mutant phenotypes, the exoskeleton of the larvae is called a cuticle, and it's made up of chitin. And there are some stereotypical structures in this exoskeleton. For example, at the tail there are tail structures, and at the head there are head structures, and it is also possible to tell the different segments by the denticle belts, the shape and placement of denticle belts on the cuticle. And denticles are these hair that stick out of the exoskeleton, and you can determine the segment by looking at the pattern or arrangement of these denticles. So here we have thoracic segment 1, thoracic segment 2, thoracic segment 3, and then A1 through A8, the 8 abdominal segments. Before we proceed further, we also need to understand a special kind of mutant that can arise during development. So let's say normal mutants are of this sort. Let's say we have M is the mutant allele, and plus is the wild type allele. Then if we take a het female and cross this to a het male, then we would get a quarter homozygous for M and 3/4 having at least one wild type allele. Now, most developmental mutants are recessive, so only these quarter homozygous M individuals will have the mutant phenotype, whereas the 3/4 individuals having at least one wild type allele will have the wild type phenotype. And the female here in the cross is the mother, and these individuals here, the progeny, they are zygotes, and it is the zygote who has both the mutant genotype M over M and also the mutant phenotype, whatever mutant phenotype it happens to be. For example, if this were ultrabithorax, then the mutant phenotype would be having 2 pairs of wings. So these normal mutants, we can also call them zygotic. Mutants, since the zygote shows both the mutant genotype as well as the mutant phenotype. Now, there is another class of mutants in development which are known as maternal mutants. And in maternal mutants, the mother has the mutant genotype, but the mother's phenotype is normal, and it is the progeny that have the mutant phenotype. For example, if the mother is homozygous for M, and we cross this female with a, let's say, homozygous wild type male, the progeny are going to be all hets, And so their genotype is not homozygous for the mutant allele. Nevertheless, they will all have mutant phenotype. So the mom has mutant genotype, and the mom is phenotypically wild type, and it is the progeny that have a mutant phenotype, even though the progeny's genotype over here is not a homozygous for the mutant allele. And so in maternal mutants, it is the mother's genotype that determines the progeny's phenotype. And given that these are all recessive mutants, if we were to do a cross of a het female with a het male, we would get 1/4 homozygous for the mutant allele and 3/4 having at least one wild type allele. Nevertheless, all progeny would be wild type, since the genotype of the mother is wild type. So once again, in zygotic mutants, and these are the kind of mutants that we have dealt with so far, the individual, the zygote, has both the mutant genotype as well as the mutant phenotype. But during development, there is a new type of mutant that's possible, where it's the mother who has the mutant genotype, and that results in a mutant phenotype for the zygote or the progeny. And if the mother has a wild type genotype, even if the zygote has a mutant genotype, the phenotype will still be wild type. One important type of mutant that was identified through this mutant screen was in fact a maternal mutant. And the name of this maternal mutant is Bicoid, abbreviated as BCD. Now, if we compare the larval cuticle pattern of an embryo produced by a mother who is homozygous for the mutant allele of Bicoid with an embryo whose mother is wild type, we can see that the entire anterior half of the embryo, so everything anterior of abdominal segment A5, is missing. So you have A8, A7, A6, A5, and then this part is actually a mirror duplication of the tail structures. This mutant phenotype suggests that the wild type allele of Bicoid specifies the anterior half of the embryo. And how does Bicoid do this? So what is the function of Bicoid became clear when the gene was cloned and its expression pattern was determined by immunolocalization. And Bicoid, as you would expect from the mutant phenotype, is actually expressed in the anterior half of the embryo. In fact, it is a gradient, so there's high levels of Bicoid in the anterior, then the level of Bicoid protein, and therefore the amount of Bicoid available in each cell, reduces progressively as you move towards the posterior. It also became clear why Bicoid is a maternal gene, and why it is the the mother's genotype that determines the phenotype of the progeny, because this protein is actually produced from translation of mRNA that is present at the anterior pole of the embryo. But this mRNA is not actually produced by transcription in the embryo, but by transcription that occurs in the mother during gametogenesis, during oogenesis. And it is the mother that deposits bicoid mRNA in the anterior tip of the embryo, and that mRNA is translated into protein, and the protein diffuses out from the head to the tail, from anterior to posterior, progressively reducing in concentration. Another type of mutant that this mutant screen revealed was called a gap mutant. And these were called gap mutants because the larval cuticle of mutant embryos, and these are zygotic mutants. So it's the genotype of the embryo is homozygous for the mutant allele, as well as showing the mutant phenotype. And the mutant phenotype is that big chunks of larval cuticle were missing in mutant embryos, as you can see over here in this mutant embryo from for a gene called cruple, where everything from the first thoracic segment to the fifth abdominal segment is missing in the mutant embryo, so that you have A6, A7, and A8 remaining in the mutant embryo. Similarly, there's another gap gene called KNERPS, where you are missing segments from the second abdominal segment to the seventh abdominal segment. And so there are big gaps in the segments that are present in the mutant embryos. And once again, just like with Bicoid and the Hox genes, visualizing where the proteins encoded by these genes are expressed in the embryo can provide insight into what these genes do. And if you look at Krupel's expression, it's expressed in this broad domain in the middle of the embryo, and it's clear that Krupel is specifying the identity of these cells that are fated to become thoracic segment 1 to abdominal segment 5. So Bicoid, the maternal gene, it divides up the embryo very roughly into an anterior half and a posterior half by having high expression in the anterior that progressively reduces as you go towards the posterior, whereas Krupel further refines the position of cells by locating them in this domain about 20 nuclei wide in the embryo, and so do other gap genes as well. A third kind of mutant that was observed in this mutant screen had alternate segments missing. So for example, if you look at this mutant called even skipped, it has the first thoracic segment, the third thoracic segment, A2, A4, A6, and A8 missing from the embryo. Whereas another mutant called paired had the naked cuticle, so the cuticle that doesn't have denticles, missing from alternate segments. Nevertheless, all the pair rule genes, as they're called, lack alternating segments, but different alternating sets of segments are missing from different mutants. Now, once again, insight into what the pair rule genes can be gained by visualizing where these genes are expressed. And if you do immunolocalization experiments, for example, with even skipped, you see that it is expressed in 7 stripes, creating 7 or 14 alternating light and dark areas in the embryo, and thereby giving the basic pattern of 14 segments that is observed in both the larvae and the adult. And the last class of mutants did not change the number of segments. So the number of segments is the same in the mutant as well as the wild type larvae. What was missing was parts of segments. So here in this mutant called gooseberry, you were missing the naked part of the cuticle of each segment, and instead of having the naked part was replaced by a mirror duplication of the denticle belt in each segment. This can be seen better in this close-up of the cuticle pattern. So here is a wild type pattern, and you can see this typical pattern of the denticles, where you have it's narrower towards the anterior and broader towards the posterior, and then you have this naked cuticle that does not have the denticles. In the mutant, you don't have any naked cuticle, so now you have denticles where you previously didn't have them, and the new denticles that have appeared are a mirror image of the pattern of the denticles in that segment. And therefore, the segment polarity genes do not cause a change in the number of segments, but they change the polarity, which is they change which part of the segment is anterior, the closer to the head or pointing towards the head, and which part of the segment is posterior or closer to the tail. And once again, when you look at the expression of these segment polarity genes, you find that they are expressed in stripes, 1 per segment. So there are 15 stripes of one segment polarity gene called engrailed over here, and it's expressed in the anterior part of each segment. Therefore, the combined if you combine all these experiments, both the genetic experiments with the mutant phenotypes as well as the experiments of visualizing where these genes are expressed, you get a picture of basically a coordinate system for the embryo. So you can liken this to the addresses that the postal service uses to deliver our mail. Bicoid, for example, tells cells whether they are in the anterior half of the embryo or the posterior half of the embryo. So that this would be something like a GPS system telling us whether we are in the northern hemisphere or the southern hemisphere of the earth. The gap genes then tell cells whether the cell is located in some broad domains about 20 nuclei wide. So this would be like having an address which puts you on a particular continent or country on the earth. The payroll genes, they tell a cell which particular segment they lie in, and this would be like in an address specifying what state you are in. And finally, the segment polarity genes tell you whether you are in the anterior part of a segment or a posterior part of the segment, and this would be like saying which city you are in. And therefore, the anteroposterior segmentation genes, they determine the body plan of the embryo, they determine all the segments of the embryo and the larvae and the adult by establishing a coordinate system where the expression of the proteins, which cells, which part of the embryo these proteins are expressed in, progressively narrows down the location of a cell. The maternal genes, such as Bicoid, tell cells whether they are in the anterior half or the posterior half of the embryo. The cap genes narrow it down further to a location, about 20 nuclei or 20 cells in width. Payroll genes then specify which segment they are in, and the segment polarity genes tell cells which part of the segment, whether the anterior part closer to the head or the posterior part closer to the tail, they are in. And therefore, cells know their position in the embryo by looking at the combination of which anteroposterior segmentation genes are being expressed inside them.